Good morning. I'm Gula Lifshitz, and it's now my pleasure to introduce our next participants. We have Dr. Nathan Jorgensen, CFO, and Dr. Tirtha Chakraborty, CSO of Vor Biopharma. Welcome to the both of you. So the format for this session is roughly a 25-minute fireside chat, and if audience members have questions during the session, feel free to type them into the question box, and we'll try to ask them on your behalf. So to start off, could you please give us an overview of Vor and its manufacturing processes for your HSC and CAR-T programs?
Yeah, thank you, Gula. And we're excited to be here at the Chardan conference.
Thanks.
So what is Vor? That's the first question. So we're a cell and gene therapy company focusing on AML, and it's a pretty unique approach. We have two parts, and we'll get into it a little bit, but we really want to focus on our manufacturing today. So one part is we take transplants, which is the standard of care for AML, and we delete off an antigen. And why we do that is to create a cancer-specific target so we can treat AML. And that's really what's missing in AML is a really good target. And that's going to enable dosing of Mylotarg post-transplant, which is our lead program. But what we're really excited about, it's also going to enable CAR-Ts in AML. And that's going to really change the game in AML and hopefully change outcomes for patients. So we have invested on our in-house manufacturing, but we use both processes, both in-house manufacturing and a CDMO, and we'll get into that. And the last thing I'll mention is we have $137 million in cash. It gets us into the second half of 2025. In the second half of this year, though, we'll have readouts from our two clinical programs. First, our Trem-cel program, which is our CD33 deleted EHSC, or engineered bone marrow transplant. We'll have additional data there, and we'll have the first data from our CAR-T, VCAR33 allo, which is our CD33 targeted CAR-T. And finally, what we may have is a cross-enrolled patient. So the patients from Trem-cel, which have the deleted CD33, can cross-enroll into the CAR-T trial. And if we get one of those patients, that'll be very important proof of concept to the entire platform. So it's going to be exciting second half of the year. So we can go to the next slide and dig more into manufacturing. So I'll let Tirtha talk us through our manufacturing.
So we are located in Cambridge, Massachusetts, and we have 3 floors for this company of ours. And one full floor is dedicated to process development and GMP manufacturing. We have 4 clean rooms, which are completely ready to go. Actually, we are using them on a regular basis for VCAR33 manufacturing. And one of the best things, as it's shown on the right side of this slide, is the seamless integration of research, process development, analytical development, manufacturing, and quality in the same facility. And this is remarkable because I know the CDMO discussion would come up over and over again. And this oversight, starting from research all the way into making a product, is extremely critical for a product as complex as not just our product, but any cell therapy product. And that is not commonly seen. And we've made sure everything is under the same umbrella. So this is our manufacturing process. The whole part of the transplant manufacturing actually starts at the collection site. When we take HLA matched donors to the patient and then apheres their material by mobilizing their cells from the marrow, the hematopoietic stem and progenitor cells, which come out from the marrow into the bloodstream. And then we collect the blood, purify these cells to have CD34 enrichment, genome engineer them to lose the antigen that we are interested in deleting, that is CD33 in case of Trem-cel, and then take it all the way to the patient at the clinical site where they're infused. For the CAR-T, it comes out of the same donor, but the other fraction, which is not 34 enriched fraction, then we do CD4, CD8 positive selection to isolate the T cells, transduce them with the vector, lentivirus in this case, in a much more compressed timing of transduction and activation, which maintains the quality of the cells, which have been repeatedly scientifically proven that how the cells look when you infuse it into the patient really matters. And then we inject that into the patient. This happens after the transplant has taken root. Now, one important thing to keep in mind, while we call this allogeneic, which is truly allogeneic, but it is not universal off-the-shelf allo. So this is a common confusion, and we want to be upfront about it. That is HLA matched allo and not universal off-the-shelf allo. As a result, we don't have to over-engineer these cells. That's one of the concerns in the universal allo field, which is not a problem for us. Nathan, you would like to add anything here?
I think that's exactly right.
So you want to take this, or you want to?
Yeah. So I know you're going to ask us questions about this too later, Gula. The advantages and disadvantages of using in-house manufacturing versus contract manufacturing. So currently we use both. And so for Trem-cel, we're using a CDMO. And the decision for that was based on speed. So we could get there faster going to a CDMO. And obviously CDMOs typically can scale as much better. However, what you do lose is some control. So when we went to manufacture our CAR-T, we had a bit more time. And so we brought it in-house. And so we could co-local that with our PD team and help develop it in-house. At some point, we could then transfer that to a CDMO, especially when we move forward and start manufacturing our Trem-cel in full. But because of our process, actually, there's not cost savings by using a CDMO. When we go to a CDMO, what we have to do is we have to train them, we have to buy the equipment, and our process, as Tirtha was just explaining to you, is essentially a benchtop process. So we have to put that equipment in there and to train the people on it. And so I think our break even is actually a very small number of patients in terms of costs and in-house versus going ex-house or external. So I think that's it for our question, for our slide deck. So we can switch it over and start with questions. Yep.
Great. I really appreciate that overview. So maybe let's dive in a little deeper to some of the things that you just talked about. So again, you were just talking about how you've invested in building out your in-house manufacturing facilities. Can you expand a little bit on how that's important to your strategy and how that has helped you advance your programs?
Yeah, so our process is very unique, and I think you follow the space closely. Manufacturing is actually one of the biggest risks to a cell therapy company. So it was important to control it and really understand our process. When we go up or do a run at our CDMO for Trem-cel, like we send people, which is not unusual, but send people in plant, and we're always close and on top of what's going on, because these are live cells, and we will talk about this later as well. These are live cells that we're manufacturing into a product. And so you can't just go off a simple template. It's a dynamic process, which you need experienced people. And that's why it's very important to have a CSO involved in the process, like from soup to nuts. And so that's why we're very close to the CDMO when we make that decision. Of course, it's a risk-based decision about whether you want to invest in going to CDMO or go in-house. Either one, but they both have their pros and cons, as we discussed.
So I guess just you mentioned doing the CAR-T in-house and then the Trem-cel with the CDMO. Can you expand a little bit on why it made sense to do that with the CDMO, and also whether there's any components of just general processes, like whether it's process development or some elements of analytics or things like that are more advantageous to do with the CDMO versus internally, and kind of just in general how you've approached the CDMO decision on the Trem-cel side and longer term.
Yeah, as I alluded to earlier, it was really a time-based decision with Trem-cel. And so we had additional time with the CAR-T. The decision will change as we go forward, and we're going to scale. At that point, it may make sense to go back to a CDMO who can be much more flexible in terms of when we become commercial. And so we're going to evaluate each, I guess, decision individually, because it's not straightforward. In terms of cost, it's not straightforward either. Each time we have to go negotiate with the CDMO, we have to, I guess, partner with them to determine who's going to pay for what and how it's going to look.
I'd like to reiterate Nathan's point, Gula, the timing. We went to clinic with Trem-cel significantly before we went to clinic with CAR-T. And therefore, it was important for us, while we were building our own manufacturing facility, not to delay the clinical timeline. And that logistics is behind it, as you said. So.
Got it. That makes sense. So in the genome edited HSC space, there has been some discussion regarding the risks of off-target editing, things like translocations or various unintended on or off-target modifications. So can you talk about how you're approaching these questions and what you've seen with your processes in TrendCell or any of your other programs?
Yeah, just in the way of background, so the Cas-JV or Exacell was a product that I developed at CRISPR Therapeutics. I was their head of hematology. So the first time ever when these discussions ensued, I was in a room with the examiners from the agency, with the FDA team. And those are the first time ever these discussions came up regarding a stem cell, because these cells are going to be with you forever. And we learned a lot. And through that process, what we created at VOR, we made it way more stringent than even what I had done with my team at CRISPR Therapeutics, CRISPR Vertex collaboration at that time. So we're very familiar with where it came from. Now, we have, just to give you a little more technical, and pardon for getting deeper into this, but like at what level off targets are dangerous? We used to look at 3 nucleotide mismatch with the guide, and we used to feel that, okay, as long as it doesn't hit within 3 mismatch, we are fine. At VOR, we started with a higher platform of 5 nucleotide mismatch. So it's inordinately more complex. And we eliminated orders of magnitude potential more targets by doing first in silico analysis and then doing experimental cross validation of the in silico potential targets to tell ourselves that everything is safe, and then tell the rest of the world that everything is safe. And when we filed our IND, there were a few questions from here and there from the agency. Not a single question on genotoxicity analysis came back. So we built on what some of my team had already done a few years before in the field, and the agency was extremely comfortable with that. Now, one of the things that we try to do is not just off-target at molecular sense, but also biological implication of those off-targets. Is this a good place or a safe place for an off-target, or it is dangerous for an off-target? Then we try to see if there is an on-to off-target translocation, off-to off-target translocation. All these things are analyzed by us extremely deeply, and we are in a very good place regarding that.
Got it. And then maybe turning to the CAR-T program, the VCAR33 program does build on some initial work that has been done in an academic setting and using an academic process. So can you expand a little bit on the differences in the process that you've been developing in-house, and also the important features of the CAR product that you produced that I think you alluded to in terms of the T-cell phenotype?
So I'll start and I'll let Nathan add anything afterwards. So the most important thing to keep in mind that what we got from National Cancer Institute was the vector only. Okay, it is not the cells, because they have an autologous program and we have an allogenic program. But even if you look at the way they develop their program using allogenic cells, because you can't do that with autologous cells when you are putting together your IND package and so on and so forth, our manufacturing process is faster, better, and way more robust. And one of the reasons for that is that we keep a close eye on the cell phenotype. The surface marker combination of these cells have been proven over and over again in the literature that they make a big difference. If you exhaust these cells in vitro, then the cells have run out of the firepower and they can't give you the necessary mileage. And in the NCI setting, this is a bridge to transplant study. So the cells may be okay if they cannot persist there forever. In our final aim of the treatment system, as Nathan was describing in the early slide, we do it in a post-transplant environment. So the persistence of these cells are going to be absolutely critical, because you're not clearing up the system. You want these cells to be there and do their stuff. So how the cells look in terms of what is known as T-cell stemness and other ability to preserve their energy before we infuse into the patient is extremely critical. And therefore, while we started with an academic construct, the cell product is all VORs, using VORs subject matter expertise in the field to build it from there. But Nathan?
So one thing we can take away from the academic studies is this looks like it's an active construct. And as Tirtha was saying, we put it in our cells, it'll probably have more activity. So they saw activity at 10 to the 7th, we're starting at 10 to the 6th, and we think that could be an active dose. Of course, we'll have to test that as we go on this year. If that's not actually the active dose, it's a 3x3 dose escalation. We'll step up pretty quickly. Definitely in the second half of the year, we should hopefully be seeing some activity.
Great. And so maybe let's move to scaling. You talked about this a little bit earlier, but what is the current scale of your processes, either in terms of doses per month or year or any other metric that you want to give? And what can you share regarding success rate at the moment?
Yeah, so we've been very successful on both products. And I think it starts with the healthy cells. Like, we don't have problems with collecting cells. So we've had, I think it's essentially 100% success rate. So in terms of our process and our ability to scale, so if we just take one clean room for either the trem-cel, which is a pretty short process, which is like a week-long process, or CAR-T, which is actually, again, a short process. It's only like 3 to 4 days. You can put a lot of different runs through either suites. And then as long as we don't start on the same day in terms of the day 0, we can then overlap processes. So, you can start calculating out that you can, put 50 to 100 runs, if you really pack them in there into an individual suite. And as Tirtha alluded, we have 4 suites in-house. And obviously we have a CDMO we can work with, and we can expand, into additional suites. So we definitely have capacity for all of our clinical development. And then it doesn't take much to, I guess, service the first few years of commercial needs. So our process scales pretty well.
Got it. So you don't anticipate needing to change things as you hit the commercial level?
That's exactly right. And so that's part of being like a bespoke one-to-one process. It won't change. Like, we take one product in, and then we have a product go out in about weeks' time. That won't change. We'll just have to add teams and staff and suites as we go. But we won't have to add a whole lot, because, if we even get up to 100, 200, 300 doses, that's only a few suites.
Got it. So I guess continuing to think about the commercial and regulatory approval type hurdles, as we talked about over the last few months, we've seen the first regulatory approval for gene edited HSC therapy, and in the CAR-T space, we've seen expanded use of CAR-Ts in the earlier line setting. Obviously, you're not at that stage yet, but again, as you've talked about, you've thought about what it's going to take to get to commercialization. And sometimes what we've seen is, what is needed to get into the clinic is not the same threshold as what's needed from a CMC standpoint to actually get through the approval hurdles. So what do you think regulators are looking for with respect to CMC for the types of products that you're developing later on to get to that regulatory approval stage?
The good news for us, and actually it's not good news for the AML patients, is that the bar is low. So if you look in the AML space, a lot of drugs are getting to market with like 20% CR rates, 20+% on single arm trials. And so I think there's some opportunity for us to get there with probably a pretty simple, straightforward process. And then your question was more on the CMC side. So we're putting into place state-of-the-art facility, which we think, and an elegant process, which is, again, quite straightforward, using healthy cells. So I think it'll be straightforward. I mean, I want to knock on wood a little bit. The space is challenging, but we'll continue to invest and hopefully, get there and not too long a time, because after we get some data from the phase 1/2 trials that are ongoing, we want to push very quickly into pivotal and get this to patients.
And then maybe stepping back from a more big picture standpoint, you've seen the CAR-T space evolve over the past several years. You've seen the HSC space evolve, ups and downs. And how are you thinking about what learnings you can take from peers in these spaces that could help you in general, and also in particular avoid some of the manufacturing obstacles and delays that we've seen plague some peers in the space over the last several years?
So I think Tirtha is great to answer this, given that his comments on CRISPR. But the other learnings is just make sure you understand the science and the process. And again, I'm going to defer to Tirtha on that one. You got to invest in the science, understanding the cell source and the cell types, and I think that'll help de-risk the manufacturing. Anything to add?
So thanks, Nathan. Yeah, you're spot on. So there is no way to oversimplify the complexity of our drug. And as I said, it's not Vor's drug. Any cell therapy drug, any cell and gene therapy drug, it's a living drug. The issue has typically been with the extremely qualified manufacturing, task force that we can get in the field. They are not trained in cell and gene therapy as long as they have been trained in everything else. So those individuals will have to learn the complexity of the drug. There is no way to feel good by saying, oh, it's just like anything else. No, it's not. And the subject matter experts, particularly in our case, take for instance, hematopoietic stem cells for the transplant and T cells for CAR T. These are deep scientific fields. And you need to have the subject matter experts working shoulder to shoulder through process development and manufacturing. And even our quality folks are learning from these individuals. So to hold it together, doesn't matter who reports to whom structurally, but under the same roof with an extremely open and regular communication is very important. If there is a manufacturing problem, all scientific subject matter experts jump in and be in one room together to learn like true scientists do. So there is absolutely no way to avoid the fact that this is a very different drug and science goes all the way.
And see an audience question here regarding profit margin. What can you share with us regarding how you're thinking about profit margin for a therapy that involves these, let's say, two components, both the trem-cel and the CAR T?
Yeah, so the two products will be reimbursed separately. So there's a DRG for transplant, and then within that there's a carve out for the hospital for the cell source and processing. So I think that will give us some flexibility on the power and pricing power on the trem-cel side. So I think we can have typical cell therapy margins there. And then it's a short process in which we take the cells in from the donor. We will process them, and we can hopefully get them out very quickly. So I think that's going to maintain or help us maintain the COGS. And then on the CAR T side, there's already a DRG for CAR T, which we could potentially take advantage of. And so that, again, it's priced pretty nicely. And then if we have the treatment system put together where we're doing one collection, we're doing the trem-cel, and actually those T cells are the waste fraction from the manufacturing of trem-cel. And so there's a tremendous COGS advantage there. So I think we're going to be in a very good position to keep our COGS very low and in line or better than other cell therapy companies, potentially even close to an antibody.
Great. So I think we're just about at the end of our session. So I'd like to thank you both for the overview and the great discussion, and we'll see you back tomorrow for the cell therapy panel.
Yeah, we look forward to it. Thanks a lot, Gula.
Thanks, Gula.